Okay, great, thanks. Okay, so hopefully slides will be working fine. They worked in the practice run, so fingers crossed. So what I wanted to do for the second bit is actually get into some of the more molecular details of latency and so the mechanisms. And again, I'm going to talk mainly about CMV because that's my interest, but I will draw some parallels with the alpha herpes viruses, particularly again, remember from that tree, the alphas were the ones that were distinct from the betas and gammas, but actually from a molecular point of view, there's actually not that many differences. And so there's common themes across the herpes viruses. And so to start, I want to reintroduce this definition. So just to remind ourselves, and it's that everything I'm going to talk about regarding latency is this concept that the virus genome is in the nucleus, it's episomal, it's persisting, but there's not lytic gene expression or replication, there's not infectious virus production, but that genome has the capacity, doesn't mean to say it will, but it has the capacity to re-enter lytic life cycle, it can reactivate. And this is pivotal when we talk about molecular latency or even cellular latency. And so again, just to remind you about latency, what we're defining latency as is a failure of the virus to enter lytic infection. And as a consequence, one outcome of that is that the virus can go latent. But it's not a given, just to remind you that if the virus fails to go into its lytic infection, then it will automatically transition into latency. So it has to be the right cell type, the right cellular environment. But what's key is this repression of IE transcription. This the first genes that are expressed by the virus as it initiates lytic infection. And if they're inhibited, then there's a chance that the virus will go into latency. So with that in mind, most people who've worked on viral latency, particularly herpes latency, have tried to investigate the mechanisms that dictate that switch, which regulate that promoter, which is making that decision of whether we get gene expression or not. And so really it's a question of cell biology and gene expression and the mechanisms that control gene expression. And so really the most general terms, you can regulate gene expression at the level of transcription. Do you make the mRNA? Or you can regulate protein expression at the level of translation. So just making an mRNA does not necessarily mean you'll get a protein. So that RNA has to be translated. And so what this provides is multiple mechanisms of regulation, which is true of eukaryotic gene expression as well as viral gene expression. And so upstream of a gene, you have its promoter, which has DNA-specific sequences where transcription factors will bind to and regulate both through activatory or repressive signals. And these sequence-specific transcription factors are important, or the activators for recruitment of the transcription factor complex, RNA polymerase II, which then recruits the RNA, and you'll get gene expression. And so, of course, this aspect can be regulated at multiple levels. So it's availability of transcription factors. So is the transcription factor which binds the DNA available in that cell? But the other aspect of this is that DNA in eukaryotic cells is not naked. It's associated with chromatin, these histone proteins, which bind to DNA and can regulate gene expression beyond transcription factors. And so this regulation of chromatin, again, we're having is called the histone code hypothesis. Regulation by chromatin is the histone code hypothesis. So DNA in eukaryotic cells is associated with optimers of histone proteins. And these histone proteins tend to sit 146 base pairs apart on the DNA. And they're essentially wrapping up the DNA in these proteins. And so they can control the accessibility of the DNA to these transcription factors, to RNA polymerase. But what's key about these histone proteins is that they are post-translationally modified. So they can have methyl groups, phospho groups, sumo groups, ubiquitin groups, and acetyl groups attached to those histones. or covalently modified on those histones. And it's the covalent modification that's on those histones which dictates their function. And this is referred to as the histone code hypothesis, which was proposed by Strahl and Alice back in 2000. And essentially what they're saying is that if you have histones with no post-translational modifications bound to DNA, there's potentially one of two major outcomes. The first outcome is you can get acetylation of those histones. And it's the acetylation of those histones which changes the charge on the proteins. The interaction of those proteins with the DNA, the negative charge on the DNA along with the negative charge of the protein causes a repulsion effect almost. And so the chromatin opens up. And because the chromatin's opening up, it can be physically accessed by transcription factors. And so we typically associate acetylated histones bound in regions of DNA as transcriptionally active regions. However, this is a dynamic equilibrium. It doesn't just sit as acetylated histones. It can be deacetylated by cellular enzymes and then become target for a family of enzymes, histone methylases, and they can push the equilibrium this way. And if you get histone methylation, this is on specific residues within these histones, particularly histone H3K9, lysine 9, or histone H3 lysine 27. In particular, you then get extensive methylation and recruitment of other proteins to that chromatin, which all act to silence gene expression. But again, this can be reversed, so you can send it back this way. And this is essentially the basis of how genes are differentially regulated in eukaryotic cells by chromatin structure. Because these changes and these enzyme activities can respond to extracellular cues like in signaling pathways. And so when I actually was doing my PhD, this is when this idea was coming out as a concept in 2000 when it was proposed. We actually asked the question, well, herpes viruses are large pieces of DNA which are sat in the nucleus. Do they get modified? Do they get associated with chromatin? And actually, are they subject to the same mechanisms that regulate eukaryotic gene expression? Could chromatin actually regulate viral gene expression? And so what I've actually got is actually the original data from my thesis, which is actually southern blots. And so people now do things like qPCR and things like that. This is actually a southern blot. And again, we are waiting. There seems to be a delay again. I think it's my computer. I apologize for this. No, there we are. So what we have here is a CD34 cell. And the CD34 cell, remember, the CMV is a site of latency. And we know it's a site of latency if we take those cells and co-culture them with fibroblasts and look for evidence of protein expression by looking for viral infection, we see nothing, essentially. So the cells here, I'm not showing them, but they're not infected because the CD34 cells are not making viruses. That's late because they're latent. And what we know is if we go and then do an analysis where we take an antibody which recognizes heterochromatin protein 1, which is a marker of repressed chromatin, or an antibody which recognizes acetylated histone H4, which is a marker of active chromatin. We see, if we look in the site of latency, the promoter, which controls the expression of IE genes, is associated with repressed chromatin. So what it's telling us is the viral genome is chromatinized, and it's chromatinized in a way which is consistent with the phenotype of latency. So the viral genome is actually being regulated by the same mechanisms that cellular eukaryotic DNA is being regulated. But the key thing for us is if we take those cells in vitro and differentiate them into dendritic cells, we can trigger reactivation of the virus through cellular differentiation. And we can see that. So what we've now got is IE positive cells, these green cells. These are dendritic cells which have reactivated virus, and then the virus is now infecting a monolayer of fibroblasts. So it's making new viruses, and we can see that infection. What's key is that the chromatin at the promoter is changed. So you may expect that to happen, but it's telling us that there's chromatin at the promoter and it's dynamic, and it correlates precisely with the latency reactivation phenotype. And so what that led us to was a model of CMV latency. And again, I don't quite know why, what it doesn't like about my computer. But essentially what we know is that when CD30, when someone's infected with CMV, the CMV goes to the bone marrow and infects CD34 cells, and there it hits a cellular environment that is inhibitory to that promoter, the MIEP. And the reason it's inhibitory is because there are binding sites for two key transcription factors, X repressor factors and YY1. These two transcription factors are repressors of this promoter, and they are expressed at high levels in CD34 cells. And so when the DNA becomes chromatinized, these proteins recruit histone deacetylases and histone methyltransferases, drive transcriptional repression through chromatin, and you get the example of latent, the establishment of latency. And it's through silencing this promoter. In a cell which is permissive for CMV infection, you often find these transcription factors are expressed at lower levels, and you see high levels of transcription factors like CREB, NF-kappa B, AP1, which are transcriptional activators of this promoter. And so just transcription factor availability is dictating whether you're getting a latent and repressive phenotype. And what's important is that I've shown you this for CMV. We also know this is true for other herpes viruses. So similar mechanisms are at play for herpes simplex virus. It's happening in neuronal cells in this instance, and it's also happening in gamma herpes viruses. So the obvious question is why are incoming genomes chromatinized? So why is the cell doing this? What's the, is this just a host, is this a straightforward response, or is this something specific about herpes viruses? And what we know from experiments where we've looked at, where we've transfect plasmid DNA into eukaryotic cells, it's chromatinized. So the host cell just chromatinizes. If it sees naked DNA, it tends to chromatize it. So it's not something specific about herpes viruses. And it's been reported, and it's now considered to be part of the host's defense machinery. It's got DNA it doesn't recognize, it's foreign DNA. One of the first things it may do is try and shut it down. It shuts it down through the action of chromatin. Almost it's shutting it down, stopping gene expression, then it's going to decide what to do with it. And so chromatinization of incoming genomes, we found, was not latency specific. And so what we realized was, when we infect cells which we know are permissive for CMV, the genome is also chromatinized. We could detect histone proteins. But what was surprising to us is actually we could detect markers of repression on that same promoter. And so despite the fact that the virus will ultimately go into a lytic infection, if you looked early enough, you could see repressive chromatin marks on that MIEP. And so what we did was we took a chemical called trichostatin A. And what this does, it inhibits histone deacetylase activity. And histone deacetylase activity, if you inhibit histone deacetylase activity, the equilibrium will shift towards active gene expression. Sorry, it seems to have shot miles ahead. And so if you've got active gene expression, you'll see it here by flow cytometry. So we took a virus where we can measure gene expression. We infected cells at a low multiplicity infection. So around about 8% of the cells were infected with CMV. If we took that same stock of virus and infected cells in the presence of a histone deacetylase inhibitor, we got more lytic infection. So what this was telling us is that there was a large proportion of cells which were infected with the virus, but were not going on to establish a full-blown lytic infection, probably because there was a defense mechanism in the cell, which in this case is repressive chromatin, attempting to silence them. And so if we disable that mechanism through trichostatin A, we can see more infection. So even in a lytic infection, there's quite clearly evidence of host defense mechanisms, which the virus has to subvert. And so one of the hypotheses when it comes to latency is that in latency, really the virus struggles to reverse these effects, even more so than in lytic infection. Yeah.
So among the modifications that you find, we also find like the more permanent like DNA methylation, and it means that this state is kind of like inherited.
DNA methylation. So it's for the CMV genome is not extensive. The methylation on the genome is not that great. The methylation tends to be on the chromatin, more so. It's not inherited. So at the end, so when the virus replicates in lytic infection, all the genome gets chromatinized. It all gets shut down through the host, shuts it down. The virus then has countermeasures which reverse that. You get activation of IE, the first proteins. They come on and then they switch on all the other promoters by basically removing the histone deacetylases from the DNA and exposing histone acetyl transferases. But at the end, the replicating genome, and we don't know how it does this, it's stripped of chromatin. So when it's put back into the virion, it's chromatin free. And so it has to go through this whole process again. So some virus, I think adenovirus, SV40 virus, actually I think the genome remains associated with chromatins. The new chromatin, it's a new genome that goes into cell will have chromatin bound. But with the herpes virus, it strips it all off and then starts all over again. So the segue into lytic infection was just to show you that this is just a general host defense mechanism, which we think is really highly active in the establishment of latency, and that's chromatin. And so what we hypothesize is that it's a viral failure to reverse the effects of chromatin silencing initiated by the cell, which is one of the major contributors to why the virus will go latent. The transcriptional factor identity of that cell is important as well. So neuronal cells are considered transcriptionally quiescent cells compared to some cells, and it's thought that actually contributes to the HSV latent phenotype. And I talked about CD34 cells in the context of CMV, high levels of transcriptional repressors, which bind to that promoter, outcompete the transcriptional activators. And so really what we're coming to is actually latency just a failure of viral mechanisms that are important in lytic infection to antagonize the host defense mechanisms. And so does the virus default into latency as a result. So it's tempting to speculate then, was regulation of viral latency all about chromatin? Is chromatin the be all and end all of this? And it's quite clearly important, but as we know from any biological system, it's not 100% foolproof. And so what I'm going to talk to you about is almost an additional layer of complexity and regulation that herpes viruses use, which also contribute, we think, to viral latency. And this is at the level of translation. And so as I alluded to, you can control gene expression at the RNA level by chromatin or transcription factors or including the RNA polymerase. But even if you get the RNA, you can still block protein expression. And one way to do that is through microRNAs, microRNA interference, and these prevent translation of an mRNA. And what they do is they are small 22 nucleotide RNAs, which function really by two different ways. So they can bind to an mRNA directly, and then they promote its recruitment into a complex called dicer, and that causes cleavage and destruction of the mRNA. So it's a sequence-specific base recognition of the mRNA, and the microRNA will bind it. And so a single microRNA in the cell, and we have loads of different microRNAs, but one microRNA will have multiple targets, host targets, cell RNA targets. The other thing that microRNA can do, it doesn't have to necessarily destroy the mRNA. It can just, through binding, making a double-stranded binding event, actually stop the ribosome scanning across the mRNA. And so the ribosome falls off. And so either way, what the microRNA achieves is a reduction in protein expression. And what I'm going to show you is that viruses have really exploited microRNAs to their own advantage. And so what I have briefly on the next slide is a little bit about how the microRNAs are produced. And so what you have is the microRNA gene, pre-microRNA. It goes into a complex called DROSHA, which processes it. And ultimately, it leaves this 22 nucleotide RNA, which will bind to the target. And as I said, it either translational repression or through recruitment. And what we've realized is from studying herpes viruses, that herpes viruses become really exquisitely sensitive to the activity of microRNAs. And so this is a really beautiful study from Don Cohen's lab. And what they showed is that neuronal cells, sites of herpes simplex virus latency, have high levels of a cellular microRNA called miR138. And miR138 clearly has roles in cell biology. It has multiple targets. But what they realized is that this microRNA also had potential targets in a viral mRNA. So this viral mRNA here, ICP0, is the equivalent of IE-RNAs in human cytomegalovirus. It's an IE, and so if you block this, you're less likely to get lytic infection. And what they showed with a really nice experiment is if you take the wild type virus, mutate it so that this microRNA can no longer bind, that the virus actually goes into its lytic replication cycle and causes death of the mouse. So in a cell where it should go latent, you get more chance to get lytic infection. And so the microRNA is losing the capacity of the microRNA to bind to the mRNA is sufficient to change the phenotype of this virus in this mouse model. And the reason why I like the experiment is the obvious experiment you could have done is just to delete this microRNA from the cell. The problem with that would have been is that this microRNA doesn't just target this mRNA, it probably has a host of cellular mRNAs it targets as well. So what they've actually done, they've left the microRNA functional in these cells, but just removed its ability to target the virus specifically. And it quite nicely shows that the microRNA has this effect. What people tend to do in herpes virology, or some people do, is that if one virus does it, maybe another one does it. And so lo and behold, what we see is that in CD34 cells, the site of CMV latency, there's a different microRNA upregulated. This time it's miR200. And this targets I86, which is essentially like ICP0. And this again promotes the degradation. And don't worry about the data. This is just the data validating it. What it's saying is that, again, the virus is going into a cell with high levels of a microRNA, which is intrinsically at high levels. It's nothing to do with the virus. It's just intrinsically high levels in those cells. And maybe is this why these cells are supporting it? Interesting, it's not just herpes viruses. And so in a minute, we're going to get an HIV slide. And so people did the same experiment. So there's a paper in Nature, it's a long time ago, and I know that there's not been lots of follow-ups of this, and sometimes it may be a one-off. So I'd be interested to speak to someone who's actually an HIV expert and whether this is really true. But the concept was suggested in HIV as well. And essentially what they're saying is that in resting CD4 T cells where HIV goes latent, or is more likely to go latent, there is a cluster again of cellular microRNAs that bind to 3' end of HIV mRNA, and they block HIV lytic gene expression. And so again, the key is that actually, do they actually have an effect? And so what you can do is use antagomeres, and these antagomeres bind to the microRNAs and stop their function, and you can rescue viral gene expression. And so it suggests that microRNAs may play an important role in latency. So the virus goes latent in cells which have potentially or inherently high levels of cellular microRNAs. So the obvious question is do viruses copy or manipulate this in any way? If the cells, it seems like a good strategy for regulating latency and reactivation, so actually the viruses do it. And so it turns out they do. So viruses have encoded within their own genome virally encoded microRNAs. And again, this was first shown really for herpes simplex virus. So herpes simplex virus has one transcript it expresses during latency, which is called the LAT. It's a latency associated transcript. And it generates this 8 kb transcript, which is subsequently processed into a 6.3 kb, and then this smaller 2 kb transcript. But it's a single locus. And no one's really ever worked out what this transcript does. They know it's expressed. It seems to have a role in the establishment of latency, although that's still argued about even to some extent. But what Brian Cullen's group showed is that you could identify potential microRNAs which could be processed from this larger mRNA. They showed that they indeed existed. And then when they looked at them, they realized that a number of them mapped as antisense as regulators of ICP4, ICP0. And so would actually regulate those IE genes. So the virus itself was encoding a microRNA, which was trying to shut down IE gene expression. And so these microRNAs are antiparallel, so ICP0, ICP4. And of course, you can imagine how mechanistically that would work. And so mechanistically, they inhibit viral gene expression and would drive latency. Of course, this is happening after the establishment of latency because the virus has to establish latency and then starts expressing this transcript. And so it's not necessarily all important for the establishment, but it's probably important for regulation. And again, CMV has a microRNA that does the same thing. So CMV has a microRNA UR112, which targets IE1, which is the other IE protein. Yep.
So, question for clarification.
Yep.
So the microRNAs, are they encoded by the virus?
So these ones are, yes. The HSV and this one are virally encoded. So the original ones are cellular ones, which are there inherently high, and they repress the virus, and they probably contribute to the establishment of latency. But now what the virus has also evolved is own microRNAs, which it expresses during latency, long-term latency, which are contributing to silencing of these IE proteins.
So the idea is that at some point, viruses obtain microRNAs from the host.
Yeah. Well, they don't necessarily come from the host. They just evolved the sequence. They're not necessarily host derived.
Okay, so there's
yeah. Well, the seed sequence recognizes the viral RNA. These viral microRNAs don't just target viral RNAs; they also target host functions as well. I don't think they've acquired them necessarily from the host. They just, yeah.
These viruses have microRNAs that are being expressed inside the host, and it's determining whether or not the host has a latent.
Well, it contributes to, I don't think it determines whether the virus actually goes latent. What it does is that once latency is established, of course, the virus could always try and reactivate or is trying to could express gene expressions. What these microRNAs may be doing is helping to keep silencing in that region in latency. And the question is why would it want to, what would be the benefit? And it's probably immunological from this point of view. I'll come on to that. So that's what they are. That's what they're thought to be doing. So if you look at the summary of the microRNAs, what we have is cells that are sites of latent infection encode high levels of these microRNAs, or microRNAs that specifically target viral lytic gene expression. So the virus is hitting a cell that is perhaps more conducive to latency. But then these viruses have evolved and actually started to encode their own microRNAs or evolved to encode their own microRNAs, and these also can inhibit viral gene expression. So the question I would put out is why bother? I've just said that chromatin regulates viral latency and viral gene expression. And I think this comes back to the idea that biological systems are not 100% clean. And so chromatin breeds, and so it will spontaneously or stochastically, for want of a better word, will change and become an active region. So you might get a bit of transcription, but not necessarily sufficient transcription for reactivation, but some transcription where you might get protein expression during latency. And so the hypothesis is what these microRNAs are maybe doing is mopping up that little bit of noise in chromatin. And by mopping up that noise in chromatin, you're essentially stopping that latently infected cell by expressing just a little bit of IE protein. Not sufficient IE protein to reactivate, because there's a threshold, the amount of protein you probably would need, but enough IE protein that might expose it to the immune system. Because remember, as I said about CMV, we have, in particular, we have 10 to 15% of our T cells circulating looking for CMV infected cells. And one of the major targets is IE1 or IE72. And so you can imagine a latent virus, a CD34 cell, just as a little blip of IE RNA, that makes a protein that gets loaded into a class 1 or a class 2 MHC complex. And a T cell suddenly recognizes it and can kill the viral infected cell. And so the hypothesis is that these microRNAs are stopping that from happening in latency. It's a belt and braces approach, as I was saying, I guess in the UK. It's chromatin's doing the bulk of the work, but microRNAs are just stopping that extra, that little trickle through of RNA. And you can see how evolution that'd be advantageous. A virus that does have the microRNA is less likely to get seen by immune system and get eliminated as a latent infection. So there's a clear advantage to that virus. Yeah.
Sorry, just out of curiosity, the other targets with microRNA that have you find any that may redirect the cell to differentiate, as you suggested there?
There is. So there's a paper from Jay Nelson's group has really led on this in CMV, and they've identified that there is a microRNA which regulates cell differentiation. So we'll drive or will control, so not necessarily drive, but will control CD34 cells and drive them in the myeloid lineage. So it's, and the thing I like about microRNAs is that they're sort of tweaking the system. They're not necessarily really broad acting. Like if you overexpress a protein, you can have quite broad effects. I think microRNAs are just tweaking. I think that's what the virus is doing. It's just tweaking things so everything's just right. So yeah, it has got microRNAs to do that as well. It has microRNAs which are also important in immune evasion, which I've not talked about here. And it's not, I think what we're trying to get away from in herpes virology and latency is not binary. It's not latent or not latent. There's gray area in between, and everything that the virus, so the virus may go latent, and then there's a lot of functions which are just making it more efficient and just making sure it's optimal. But if you lose those functions, the virus doesn't necessarily just go full-blown lytic. It just might be less efficient at latency, and that might be sufficient in vivo to give it a disadvantage. But yeah, but they're doing all sorts, these microRNAs. Okay, so I want to finish a little bit about, start to talk about a bit more of our research interest. And our research interest, and this is why I was banging on about latency being defined by reactivation, because we work on reactivation, so I want to make sure it's the most important thing. But what we're interested in is, what I've told you so far is how all these mechanisms, all these ideas of how the virus goes latent. But if the virus never escapes from latency and never reactivates, then who cares to some extent? So I'm going to go back to the original slide that I showed you, which had the chromatin on. And to remember, and when the CD34 cells with the viral genome in, they have a repressive chromatin. As the cells differentiate, that chromatin changes, and there seems to be a longer delay here. Okay, and so just remind you. So we see this happens, but some people might say, well, this is a self-fulfilling prophecy because you've got viral reactivation. But surely, if you think chromatin is important, of course it changes. It's got to change because this is what's regulating viral gene expression. So the real question we wanted to understand is not necessarily that this happens, but how does it happen? Why does it happen? And so by way of analogy, if you were to take a CD34 cell and take a look at the chromatin, the promoter structure of every gene, 20,000 genes in the eukaryotic, in the human genome, some would be acetylated chromatin, some would be methylated chromatin, because not every gene is being expressed. But when a CD34 cell differentiates into a dendritic cell, not all those repressed promoters will automatically activate. Something has to happen. And so the real question is when a cell differentiates from a dendritic cell, what signals occur that instruct the chromatin at that specific promoter to go from a repressed state to an active state? And what we know is from studies is that we have to differentiate the cells. So what is so special about taking a CD34 cell and making it into a dendritic cell? And linked to that, what specific signals are important for reactivation of dendritic cell, which for some reason will not trigger reactivation of the CD34 cell? And the hypothesis we went with a while ago is actually what dendritic cells are very good at is responding to inflammation-induced inflammation and induced inflammation-induced signaling. And the major immediate early promoter, that key promoter that dictates latency and reactivation, essentially looks like an inflammatory promoter. It's full of NF-kappa B, natural NF-kappa B binding sites, Krebs transcription factor binding sites. It looks like a eukaryotic inflammatory promoter. So we wonder whether it's just behaving in the same way. And so to test this, did a low throughput screen. Essentially, I went in the freezer and pulled out lots of different cytokines and what are potentially inflammatory. And we took our model of CMV latency, and that will be here again. So I thought, well, why is my thing not working? I don't know why it doesn't like my computer. Okay, tomorrow we'll use a different computer. Yeah. So cytokines are essentially immune of normal. Well, not actually, that's not true. They are little proteins secreted by cells, often immune cells, which then signal either locally in say a site of infection, but actually can signal throughout the body and promote cellular migration. So, for example, if you're infected at a mucosal membrane, one of the first things that might happen is you get an induction of interleukin 8, which is a cytokine. Interleukin 8, one of the things it does, it sends a signal and tells immune cells to come to a site of infection because you're needed. It's basically an SOS call. It's one of the things. Cytokines also drive cellular differentiation. And what they do is they bind to receptors on the surface of cells and initiate signaling inside that cell. Okay. What we know is that inflammation, so basically high levels of cytokine production, interleukin 6 is a classic, interleukin 8 another classic, are really high in certain inflammatory settings. So for example, graft versus host disease and transplantation, we get really high levels of these cytokines. In sepsis patients, often we'll get high levels of interleukin 6 in these patients. And it's just a marker of infection in some respects. And what we know is that CMV is more prevalent in patients with highly inflammatory situations. And so the simple model is we take CD14 cells out of somebody's arm, infect with a virus, differentiate into immature dendritic cells in the lab using cytokines, and then we trigger reactivation with any of these cytokines and look to see whether we can see anything. And the key thing to take home from this is that interleukin 6 is an important trigger of CMV reactivation. Now, LPS is up here as well, but actually what we went on to show is that LPS induces interleukin 6 from the cells, which then feeds back and triggers reactivation, but I haven't got time to show you that here. So the virus is responding to high levels of interleukin 6. What we went on to show is that when the dendritic cells are stimulated with interleukin 6, they trigger reactivation, and that reactivation is triggered through an internal signaling pathway. Now, the details of the signaling pathway are important. It's a pathway that's called MAP kinase pathway. So there are multiple different pathways in the cell that can respond to cytokines. We have the MAP kinase, the P38 MAP kinase, and the JNK MAP kinase. These are really important inflammatory signaling pathways in dendritic cells. If we take an inhibitor, and I apologize for the numbers, but we take an inhibitor, we massively reduce CMV reactivation. What we do is we're measuring viral gene expression. The ERK inhibitor really kills viral reactivation. And so LPS and interleukin 6 use MAP kinase signaling. And the reason we thought we were interested in that is that interleukin 6, when it signals to MAP kinase, has been shown to activate Krebs responsive genes. And remember, Krebs is what I told you about is a transcription factor which binds to the major immediate early promoter, the CMV promoter. And through activation of the Krebs transcription factor, you get by. And so what we hypothesized, because the virus has multiple CREB binding sites, it's a transcription factor that's pro-transcription, maybe it's important. And what we did was we showed that CREB was important for reactivation. So what we actually did was we deleted the CREB binding sites out of the virus, and we saw that the virus had a defect in reactivation. So if we remove the ability of CREB to bind to the virus, we lose the phenotype. That's all very well and good, but these are pretty pleiotropic cytokines, and activities. HERpMAP kinase signaling is active in every cell. Most cells respond to interleukin 6. The CREB transcription factor regulates the expression of 4,000 genes. So if we wondered if we start to pull apart the nitty-gritty of it, will this give us any clues about why interleukin 6 activates ERK and triggers reactivation in these cells? Essentially, if you want to phosphorylate a protein, you need a kinase. It's an enzyme that phosphorylates proteins, and signaling pathways work through kinases. Protein X phosphorylates protein Y, protein then Y, then that phosphorylates protein Z, and eventually you get to the end. What we identified was that this protein called miGen stress activated kinase was important. And we were pleased to see that because it allowed us to answer the question that I posed right at the start of how do you see the change in chromatin occurring? And the reason we see the change in chromatin occurring is because that miGen stress activated kinase doesn't just target CREB. It also has a really no other important target. And the other important target of this kinase is histones. And so as well as phosphorylating CREB, it phosphorylates histones. And that becomes important because histone phosphorylation is the signal that tells repressed chromatin to go to active chromatin. And so it's the activation of that signal which drives that reactivation phenotype. So CREB binds the viral promoter, recruits MSK, and MSK, then because it's in the proximity of those histones at that promoter, will phosphorylate that promoter. What that phosphorylation does, it's on a residue next to the lysine 9 residue, which this recognizes. By phosphorylating it, it destabilizes that, and you get gene expression. It opens up the chromatin. And so what we've finished with is a model of CMV reactivation where the virus is responding to essentially extracellular cues. And what's key is that this is the latent promoter. We get activation of HERpMAP kinase MSK. It binds to the CREB transcription factor and phosphorylates it. It then phosphorylates the histone, destabilizes interaction. An unknown mechanism recruits unknown at the time, recruits the histone acetyltransferase activity, and we get gene expression and reactivation. The key thing is, this is not just CMV. All herpes viruses have the same problem. How do you get from a silence promoter to an active promoter? And what became clear is from studies in KSHV, actually you would use the same mechanism, use the same CREB transcription factor mechanism, and MSK. It's slightly different in alpha herpes viruses, but they do the same thing. They phosphorylate the histone. They just use a different kinase and use junk kinase. And the key thing about junk, it's a stress activated kinase. So if you're stressed, that kinase gets activated in your neuronal cells. And what we know about herpes simplex virus reactivation, it reactivates in response to stress. And so that's the stress has been on an organism level, that's the molecular link in herpes simplex. That kinase gets activated. So that's all very well and good. Okay, so you get activation and you get activation gene expression. But if we go back to the CMV story, so CD34 cells have to differentiate into a dendritic cell. Then when they differentiate into a dendritic cell, you get reactivation, but it's interleukin-6, which is a pleiotropic cytokine. That'll act on every cell which has got an interleukin-6 receptor. It's not a specific cytokine. Anything that can respond to interleukin-6 will be important. HERpMap kinase is active in all cell types. So what is it about a dendritic cell that if you add interleukin-6, you get reactivation, but if you take a CD34 cell and add interleukin-6, the virus won't reactivate? What is it about this pathway in dendritic cells that makes it functional, but doesn't necessarily make it functional in CD34 cells? So we wondered whether can we get at this through phosphoproteomics? What else is changing in the cells alongside? So what we tend to do in biology, we have our signaling pathway of interest, and we just focus on that, and that's our interest. But nothing happens in isolation in the cell. It's all sorts of things going off. So what we really needed was a cell type that doesn't support CMV reactivation in response to interleukin-6, which looked a lot like a dendritic cell. Fortunately, I had a student in the lab who'd just been looking at this model system, which essentially allows you to take monocytes and make two different types of cell. So we make monocytes into classic, we turn monocytes into classic dendritic cells, and that's what we use in the lab normally. What Margaret showed is actually you can take monocytes and make them into a slightly different type of dendritic cell, which is called a monocyte-derived Langerhans cell. It looks like a dendritic cell, it's a bit more macrophage-like in its phenotype. And so what we know is, very clearly, we have this very clear discriminatory phenotype. One will reactivate in response to interleukin-6, but one won't. And so the first thing that we said was, oh, okay, so what do they, how do they respond? Can they respond to interleukin-6? And what I'll show you in a minute of the data is that both these cell types express the interleukin-6 receptor. So they both got the receptor for the cytokine on their surface. So the theory, and we can see that these cells that don't reactivate do phosphorylate ERK, that kinase, which is really important. So the cells can respond to interleukin-6 absolutely fine. So a lack of reactivation was not a failure to respond to the signal. There's something else going off at the same time. So the real question was were they responding differently? And so to get at this, we took cells, monocyte-derived DCs, monocyte-derived Langerhans cells, hit them with interleukin-6, and they did phosphoproteomics and looked at the complexity in the signaling pathways. We basically looked for things which were the same and looked for things which were different. So what proteins get activated in a cell type that reactivates versus a cell type that doesn't? And what we saw doing this approach is that the two biggest changes were in a second pathway. And that second pathway was activated to much higher levels in our dendritic cells. Very brief, I don't label this one, but essentially we took inhibitors of this pathway, which is the SARC family kinase pathway, and they block CMV reactivation. So now we had two pathways which were important for CMV reactivation. One was the SARC family kinase over here, one was HERpMap kinase pathway over here. Okay, so they both block reactivation, but are they completely separate, or actually are they working together? And what we went on to show, and I don't want to show necessarily show the data, is what we wanted to understand is what or how they are linked. Are they linked together, or are they separate? And so what we found out was, and I'm not going to go through the data, I'll just hopefully show you the model in a minute, is that interleukin-6, as well as activating HERpMap kinase signaling, is also activating SARC family kinase signaling in these dendritic cells to very high levels. So interleukin-6 is activating ERK, as I've said, activates MSK. We get the first stage of CMV reactivation, which is this histone phosphorylation. The chromatin then reaches a decision point. They either go on to full-blown activation, i.e. acetylation, or reverse back to repression. And we know chromatin will do that when it's phosphorylated. The phosphorylation is a transient event. What the SARC family kinase signaling was doing was at the same time recruiting the histone acetyl transferase. So it's a double hit. And the way we think about this is that when you start thinking about signaling, you can't think about signaling as linear modules. And the best way I've always told the students in UCL in London is to think of the tube map and how all the tube lines all cross over each other. So if you actually inhibit the Piccadilly line, which is one of the tube lines in London, it has a knock-on effect. If you shut the Piccadilly line, it has a knock-on effect on the Jubilee line, because all the traffic that would go through the Piccadilly line now has to go somewhere else in the system. And we think this is true of how we should think of signaling. And this is what the virus is telling us. Actually, when you inhibit one pathway, it has a knock-on effect everywhere, somewhere else in the biological system. And so to finish this aspect of it is there are a number of conclusions we can potentially draw. So herpes virus established latent infection in cells that inherently express lower levels of proteins or functions that are required for virolytic gene expression. So transcription, availability of transcription factors, for example. We think latent herpes virus gene expression is regulated by cellular chromatin. The data would point towards that. And we actually think this is, and not just us, I think many people would think this in virology in general. And actually in any form, people work with any form of DNA. If you put naked DNA in a cell, the host cell defends itself against that naked DNA. And the defense mechanism is to silence it. And it silences it through chromatin if it's not integrated. If it's integrated, like HIV, it silences it through things like Hush complex, which actually recognize it and again will just silence it. And what the host cell is doing is trying to protect itself from foreign DNA. And the quickest thing to do is shut that DNA down. And normally that works. And it would work. It shuts down the latent herpes viruses, the genomes of the herpes viruses. But it also allows the herpes viruses to persist because the genome now looks like host because it's chromatinized. So now the host cell thinks this is just another of my chromosomes. And so the DNA sensing pathway, which would recognize foreign DNA, no longer can recognize it. And that's probably how the viral genomes can persist without being recognized by the host as foreign, because they're not foreign. It's just a piece of DNA with chromatin on it. It just happens to be viral. The key thing we want to emphasize is that viral reactivation is really dependent on cellular signaling pathways. And it's different cellular signaling pathways for different viruses because they're in different cells and they've got different receptors for different cytokines. But mechanistically or fundamentally, it's the same problem. It has to reverse the chromatin structure, and it does it through, may do it through different kinases, but ultimately you get the same outcome. You get chromatin remodeling. What we think is quite nice is that microRNAs are highly expressed, cellular microRNAs are highly expressed in latent infected cells where virus will go latent, and they can mediate viral gene expression. But what's perhaps even more interesting is that viruses appear to mimic this, and they actually encode their own microRNAs. And I feel like it's a bit like a relay race almost. So the host cell is intrinsically inhibitory. It's got high levels of these microRNAs, which contribute to the silence. The virus establishes latent infection, but it needs to maintain that latency. So then it starts to express its own functions, its own microRNAs, which helps contribute to that silencing phenotype. We think that's going to be important, that silencing, because it protects it from immune recognition through adverse gene expression and protein production until the virus actually is in its cellular environment, which is optimal for reactivation. And so I put this question at the end, are the viruses just passive observers of this? Clearly, this data here suggests they're not. And what I will show on tomorrow is actually the viruses really are not passive. And so sometimes, you know, this is about dormancy. Viruses which are latent are not dormant. I would argue they are just as involved in regulating the host cell environment during latency as they are potentially during lytic infection. In fact, it's more nuanced than that because this can be quite long-term effects. So herpes simplex virus can establish latency for years in a cell. And so I think that's the last slide. So I'll just finish there. Thank you.